The next speaker is David Fenyo from the Rockefeller University, and he's going to talk to us about MRM analysis. Thank you, David.
Thanks, Mike. Yeah, so I'm going to present a method to detect interference in multiple resonance monitoring. The background to this is that when you do apply MRM analysis to quantify low abundance proteins, you sometimes do get interference from other components of the sample. So I will present a very simple model for how interference happens, and then use computer simulations to query this model and see what the properties are, and then finally apply the method to experimental data. So in MRM analysis, what we do is that we analyze samples separately and then select parent ions and look at specific fragment ions. And it's the intensity of these fragment ions that is used to quantify. And usually one uses a few transitions per peptide, typically between 3 and 5 transitions. To get higher accuracy, one often uses heavy synthetic peptides that are spiked into each sample. So in this case, one would look at the same transitions both in the light and the heavy peptides for all the samples. So as I mentioned, the motivation to looking at interference is that even if when you develop the MRM assay, you look for interference and you analyze a lot of samples, look at different concentrations. When you finally apply the methods to real samples, you can get interferences that you don't anticipate, that you never saw while you developed the assay. Also when you move the assay to a different lab, you can get different interferences. So if we look at some experimental data and to see how MRM quantitation is done. So this is data taken from the CPTAC verification workgroup study 7, which was a multi-lab study where they looked at 10 peptides, 3 transitions per peptide, over a concentration range of 1 to 500 femtomole per microliter in a human plasma background. So, and the study was done by 8 labs, and each lab did 4 repeat analysis of each sample. So these showing here is 2 peptides that show very nice linear response over the whole dynamic range for all the 3 transitions. But in a few cases, one would get interference. So, for example, in peptide 3, transition 1 does suffer from interference. And in peptide 4, we see that transition 2 suffers from interference. So how do we think about this? So we devised a simple model. So if we look at the ratios between transitions, we see that when there is no interference, we see that the ratios of the transitions are constant over the whole range for the transitions. So this is so these experiments were done with an heavy labeled synthetic peptide as an internal standard. So the straight lines here show what the median ratio for the internal standard. So we see that our transitions are very close to the internal standard. Then when we look at a case where we do have interference, we see that the ratios between 2 and 3 is also linear, because in those transitions we don't have interference. But we do have interference in in transition 1, and then the ratio deviates a lot from from what we expect. So the model that we devised is very simple. So we know from measurements what the expected ratios of transitions are. And then there is noise, so that in every measurement we're going to get deviations from the expected. And then we have interference, which is another species in the sample that has the same parent and fragment mass. So that's a positive and something that we add to one or more of the transitions. So what we do is that we detect outliers in the ratios of transitions. So what we say, we calculate a z-value for each transition. And then if that is larger than a threshold, then we say that we have interference. And this z-value for a transition I is calculated as we take the maximum of all the z-values, where we compare transition I to each other transition j. So here we have the expected ratio between transition j and I, and then we subtract the measured ratio, and then simply divide by the standard deviation. So what we just simply measure how far from the expected ratio we have the outlier, how many standard deviations from the distributions our measurement is. So if we apply this method to, now these are from computer simulations, so if we apply this to the case when we have no interference but we have noise, so we want to see how much worse we do when we try to detect interference in a case when there is no interference. So if we set the threshold to infinity, we don't have we don't do any detection. So this is just the distribution of the relative error when we don't have any correction. So here we have relative noise of 0.2, and we don't have an interference, and we have 3 transitions, each with equal relative intensity. So now if we go and set the threshold very low, so we set it to 0, so that is on the average we'll throw away every second point, because that would be considered an outlier. We see that we get a shift in the distribution, and we get the error increases. So this is definitely too low of a threshold. So what we're going to look now, we're going to look at how the centroid changes as we move up in the threshold. So this plot here shows the threshold, the threshold value, and for 0, we end up somewhere a little bit lower than minus 0.2, which corresponds to this centroid here. So if we increase and go to a threshold of 1, you see that the curve shifts towards the curve when we don't use any correction. And finally, if we go to z equal to 2, we see that we get almost the same curve as when we don't correct, except for a tail at large errors towards large negative errors. So then, now we're going to look at data with interference. So I'm going to show this on a different kind of plot, where we, on the x-axis, have the relative error before correction, and then on the y-axis have the corrected relative error. So on this plot, if we don't apply any correction, then we're going to get diagonal. So that means that the relative error before will be the same as after. And if we do perfect correction, independent of what the relative error is, we're always going to get a 0 error afterwards. So if we look at the same case as before. So now we have a relative noise of 0.2. We have interference in 1 out of 3 transitions. And again, we have 3 transitions, each with the same relative intensity. So now, if we don't do any correction, all the points will end up on the diagonal. And if we do a correction with a threshold of 2, we see that there are some points left on the diagonal, but for most, we get points around 0, and the width of this depends only on the noise. So if we look a little bit more in detail at this, what happens? So if for a threshold of 0, and now we cut out this box here and only look at the values. So this axis here is going through this here and integrating over the grayed area. So we see that now for a threshold of 0, we get a very similar distribution as for the case when we didn't have noise, when we didn't have interference but only noise. And again, when we move to higher thresholds, the curve moves towards 0. But now when we raise it, go up to 2, we see that we start getting some interference that's left over. And you can see that those are these dots left on the diagonal. And if we go to a threshold of 3, we start getting quite a lot in this left that's not corrected for. So if we move further out from the higher interferences, we see that again the curve moves in, but out here we don't have any interference. So pretty much what this means is that if we have a large interference that's much bigger than noise, we can very easily detect it. That's the principle. So now we looked at interference in 1 peak. So if we move to interference in 2 peaks, we see that, so this is the old data that I showed before, so 1 interference out of 3 transitions. So now we have a much worse case where we have 2 interferences out of 3 transitions. You see that we get quite wider distribution here, and if we look at this in detail, so this is comparing it to the case when there is no interference, only noise, and then 1 interference here, and then again 2 interferences. We see that we can do some correction even in that case, but we do get quite a wide error distribution. And probably in this case it's better just to reject the data and completely and say that if we don't have at least 2 transitions that agree and they don't have interference, then one should probably just throw away the whole measurement. Okay, so then if we go back to the experimental data, so here now I've averaged over the 4 replicates and the 3 transitions, and we're showing the uncorrected data with the blue circles, and then the corrected data with the red triangles. So these are the 2 peptides that did not have any interference. So it's good that we also then, after correction, don't introduce more error. So if we look at what the error is more in detail, so now this is the relative error we've zoomed in, we can see that for peptide 1 we get about the same errors as before and after correction. So we haven't made it worse in this case, which is good. So in peptide 2 there might be a slight improvement after correction, but it's minor. And then if we move to the 2 cases where we do have interference, so we see that we can correct quite well in both cases. And if we look in detail at what we see, so we see that the relative error is very large for the uncorrected in both cases. So if we zoom in again in both these, and then we see that we get, for the high intensities here, we get the same errors, but then for the low intensities, we remove the interference. And then in this case, again, we can get down to very low interferences after correction. So what I've presented is a method that can be used to detect interference. And then after the interference is detected, one can decide whether one will, corrects the quantitative measure for these interferences, or one rejects the whole measurement if there is too much interference going on. So the method is that first one, when developing the assay, one measures the intensity ratios of the transitions so that one knows what ratios to expect, then estimates the noise of the ratios. And this can be done with the repeat analysis, usually. But there are some problems with estimate, and this is probably the most complicated part, to estimate the noise correctly. And then the next step is that one finds and removes the transitions that contain interference, and then decides whether there is enough transitions left to do the quantitation, or that whether one should reject the whole measurement. So in this case, we had data that contained both the light labeled sample and the heavy labeled internal standard. So we can apply this method separately to the internal standard and to the sample, so that one can also detect interferences in the internal standard. And one can also use it to detect if there is ion suppression or saturation. Thank you. Oh yeah, so, and I want to acknowledge my collaborators also.
Questions, please, go ahead.
So I had a little picky question. When you had no interference, but you had noise, it shifted off of zero asymmetrically. Was that because you were adding noise to both numerator and denominator in the ratio?
Yes. Yeah, so you have, since you have noise, you're going to have outliers that will be detected. If you have the threshold too low, they will be detected as interference, even though it's just noise.
Okay, I think I get it. I think this comes up in other places, like in the significance analysis for microarrays from Stanford, and they have like a fudge factor they add to the denominator to correct for that. So.
Please, go ahead.
Is the problem that you're encountering a mass space problem, or is it an inherent lack of reproducibility of the MRM experiment?
I mean, it's definitely in these studies, some labs saw interference with the same transitions, and others labs didn't. So I mean, that would definitely be that maybe the chromatography is slightly different, and you get different peptides coming out at the same time.
So Jamie Sherman showed that there is a mass space issue, that there are peptides that are just going to be very close to other random peptides in the whole proteome. And there are others that are really unique, and so should you be looking for those rather than trying to measure the ones you can't measure? Yeah, no, but I mean, that is done in the, when people develop their assays, they look at that, and that's usually taken care of. So these are then things that, I mean, you can look at masses, but I mean, you can't predict all the post-translational modifications that you have. I mean, there's a lot of things that you can't anticipate when you develop your assay. Yeah. Okay.
Next question, please.
I was just wondering if you could comment on the option of identifying I'm making corrections versus just eliminating the transition of interference. So basically you'd have a dynamic exclusion type of approach where you drop the one that causes you the problems, retain the transitions that give you a good measurement.
Yeah, I mean, that could probably be done on the, you mean on the data acquisition? Or?
Actually, after you process the data, if you identify a measurement that you said, you can identify ones that have an interference. And so rather than applying a correction, looking at that data where you just drop one of the transitions that cause problems as opposed to applying this correction.
Yeah, no, that's what I was mentioning, that if you have too much interference, it's probably better to drop it.
Okay.
One last question, please.
Actually, I have a problem with this statement that if you have too much interference, you should throw it away and not work with it. And it's believed you're negating a whole field called multivariate calibration that is applied to other kind of spectroscopies like NIR and stuff like that, in which you never have a specific signals, but you have a bunch of unspecific signals that you work together, creating a multivariate space and calibrate on that space. Have you tried something like that?
Yeah, so, I mean, it depends on what you're looking for. I mean, if you really want to calibrate accurately, you do want to base it on a few different measurements. I mean, if you just want to have a rough measure, you can definitely use this method even if you have interference in all, let's say, 3 out of 3 transitions. And you'll get, if you apply this, you'll get a better measure of the quantity of the peptide. But it's still not going to be very good.
Well, I disagree with that, but we can discuss that later if you want. Thank you.
Okay, let's thank David again, please.